Hello everyone, welcome back to another session of RNA biology. So we were here in the previous class that we were discussing the ways in which X chromosome can be inactivated, and we saw that in embryos, until the formation of extra-embryonic layers, the paternal copy is inactivated, whereas in embryonic stem cells, both the copies, either paternal or maternal, can be inactivated randomly. So what is imprinted and random X inactivation? In tropectoderm and primitive endoderm, this is embryonic terminology, of the blastocyst, X inactivation is subject to imprinting of paternal X chromosome. This is what is called imprinting, means there is no equality given to the maternal copy and the paternal copy, or here it is biased to inactivate the paternal copy or father's copy until this stage. So that is why we call it as imprinting. It is being assigned to do so. In epiblast cell, X inactivation is random. And remember, epiblast cell is the one which is going to become the embryo, the actual organism. Other part is not going to become the organism. It becomes the extra embryonic layers, other tissues such as supporting structures such as placenta. So early X-cyst expression is exclusively of paternal origin. You can guess it, why so it is happening, why paternal copy is being arrested, because the paternal X chromosome express the X-cyst, and this X-cyst RNA will coat onto the same chromosome, and hence it will become inactive. The maternal X-cyst allele is repressed. Allele means alternative form of a gene. It's like you have every gene two copy. One came from father, one came from mother. But whether both are expressed, it depends on which gene. Any gene, I am not talking about X chromosome, like chromosome number 1, chromosome number 2, chromosome number 3, any chromosome gene. So if only one copy is expressed, paternal or maternal, or randomly anyone is expressed, we usually call them as monoallelic gene, or such expression is called monoallelic gene expression. But in some cases, both copies are expressed. Paternal also expressed, maternal also expressed. I am not talking about X-cyst, I am talking about a regular normal gene, any gene, gene A, gene B, gene C, gene D, any gene you can, like we know 23,000 genes are there in human. So any of these genes, they can either follow a monoallelic pattern and biallelic pattern. Many times this monoallelic gene expression pattern are called imprinted, like what you are seeing here in the case of the X chromosome. Until the extra embryonic layers, the paternal copy express the X-cyst, hence it is made inactive. Whereas the maternal copy of X-cyst is not allowed to express, and until the morula stage. Once it crosses the morula stage, the epiblast structure will have a randomization. That means the maternal one also will start expressing. Now it's a question of time, who wins over whom, or who answers, just like some quiz competition you would have seen, that is called rapid fire round or bell buzzer round. It's like question is free for all the 4 teams or 5 teams. So whoever ring the bell first will get a chance to answer it. So this is also something similar to that. Whichever express the X chromosome will get a chance to get into inactivation of that chromosome. So this is an article that is published in Science a few years ago, very interesting article, and epigenetic dynamics of imprinted X inactivation during early mouse development. It is published in 2004. Here they categorically show zygote is a single cell stage, it becomes 2 cell, 4 cell, 8 cell, and 16 cell stage onwards is morula, and blastocyst stage it's around 32 cell stage, and we can see that this will give rise to other structures like tropectoderm and also primitive endoderm and inner cell mass. Inner cell mass of the blastula is called ICM. So that have the stem cell characteristics. So let us see. In 4 cell stage, RNA polymerase 2 is excluded. You don't want any gene to express from the X-cyst expressing RNA. So X-cyst coating happens on XP. XP stands for X paternal. Paternal means from father. And at 8 cell stage, you have H3K4 hypomethylation, H3K9 hypomethylation, hypoacetylation, etc. So H3K4 hypomethylation and H3K9 hypoacetylation takes place at 8 cell stage. Then at morula stage onwards, you have EED, ENX accumulation. These are all two proteins that accumulate, and H3K27 methylation takes place, and you have this unique histone protein, that is macro H2A association. You can see in those X chromosome in all these cells. So this paternal X chromosome start exhibiting these features. At around blastocyst stage, you have H3K9 methylation taking place, and then you have got TE tropectoderm, and these are all happening at the blastocyst stage, and you have early replication timing, that is more than 60 cells of the primitive endoderm will show the early replication timing. That means the replication means cell replication, and of course the X chromosome also have to replicate. And the reactivation, that inactive paternal chromosome is now reactivated in the inner cell mass, because if paternal remains inactive, then there is no way the maternal will get a chance to get inactivated, because both the X chromosome cannot be inactivated. So you have to start from the scratch, just like you had a whiteboard or blackboard and you have completely written. So now you want to write more, you have to erase it completely. So this reactivation happens in the inner cell mass of the blastula, which eventually becomes the proper embryo, so that X-cyst can express either maternally or paternally. So the loss of X-cyst RNA coating on the paternal X chromosome takes place, and EED ENX association is lost, and H3K9 and H3K27 methylation is lost, so that the reviving of this paternal X chromosome happens. Now either maternal or paternal can get inactivated. So let us see X inactivation. How does this happen? X inactivation happens via X inactivation center, X chromosome inactivation center. So it happens in a locus of X chromosome. So it contains several non-coding RNA, that is called X-cyst, T6, JPX, and activator of X-cyst, and FTX, and which is 5' to X-cyst. So X-cyst we know, X inactivation specific transcript, and we also know T6, which is the antisense strand, and JPX is a non-coding RNA that is called activator of X-cyst, and then FTX is another non-coding RNA that is 5' to X-cyst. So these are all the main 4 non-coding RNA found in the X chromosome inactivation center, or XIC. And X-cyst gene transcribes a long non-coding RNA X-cyst, that is roughly 17 kilobases in humans, and X-cyst RNA is spliced, polyadenylated, but not translated, because it do not have the codons. So splicing and polyadenylation happens before it can coat onto the X chromosome. So X chromosome inactivation center, it has many genes. They are all meant for inactivation purpose. So roughly around 12 genes are there, and it can give rise to 7 proteins and 5 untranslated RNA. X-cyst and T6 are one of the 5 untranslated RNAs in the X inactivation center. On the active X chromosome, T6 expression antagonize X-cyst. That means one of them has to express first, either T6 or X-cyst. If X-cyst express first, then that chromosome will be inactive. If T6 express first, it will not allow the X-cyst to act on it. So also X inactivation center is important in counting and selecting the choice of the X chromosome. Choice usually is random. There is no preference is given to paternal copy or maternal copy. Choice usually is random, but counting takes place. Counting means cell has to be sure that they have only 2 X chromosome. What if there are 3 X chromosome? You have to inactivate 2 of them. What if you have 4 X chromosome? You have to inactivate 3 of them. Under any circumstance, allow only 1 X chromosome to function. Like I told you in the earlier class, that if such a mechanism existed for 21 chromosome, then Down syndrome wouldn't have been a problem. What if, how nice it would have been if every chromosome pair has an option of counting and making sure only 2 are expressed, then Down syndrome wouldn't have been a major problem at all. this is not easily possible because every chromosome don't have X-cyst or similar kind of non-coding RNA, or every chromosome is not as small as X chromosome. X chromosome being small, compacting and condensing will be much easy. So huge ones are very difficult to condense. With the help of one X-inactivation center, it is not that easy. So let us see in detail the X chromosome inactivation center, XIC. You can see here it is part of the chromosome itself, and this is the scale. So this much distance counts for around 10 kilobases, and the direction of various genes are also shown in arrow. Direction means which direction it is expressed, means 5' to 3'. So P stands for P0, P1, P2, these are all different promoters. What sequence elements it is going to utilize for the expression point of view, or means what are the regulatory elements that allow the polymerases, RNA polymerases to come and recognize. So in X-cyst, you have either P1 or P2 promoter that is expressed the X-cyst, and depending upon which stage of embryonic development you are in, accordingly it will use P1 or P2. And then this is going from 5' to 3', this direction, X-cyst expression. And opposite to that, you have the other strand, opposite direction, you have the T6. And also you have got TSX, BRX, CDX4, like that. Few of the non-coding RNAs are there, and then you also have XCE, and their location is been shown with this line that until where it is going. So the choice has to be made at the level of the T6 or X-cyst expression, which gene is being expressed is decided randomly, and the counting has to take place. And counting means how many X chromosomes are there. Usually a healthy female will have 2 X chromosome, and they have to, but there can be rare situation where 3 X chromosome can be there. In that situation, both of them, 2 of them will be made inactive, only one is made active or allowed to be active. So that is why counting becomes very important. So cis inactivation is mainly done by X-cyst, whereas the choice selection also is governed by X-cyst and also contributed by other downstream genes like TSX, etc. And also the counting also contributed by some of this chromosome, and the choice is also contributed by the XCE non-coding transcript also. So these boxes indicate that which genes are contributing to what purpose or what process. So 3 main things has to be taken care of. One is choice, which X chromosome should be made inactive, and then counting, how many X chromosomes are there in a given cell, and follow up with the cis inactivation. That means which chromosome express the X-cyst, that chromosome should be made inactive, and which chromosome express T6, that chromosome should be maintained active. So X chromosome inactivation center, that it can increase in the stability of X-cyst transcript, is usually brought by developmentally regulated switch in the promoter use, like we saw P1, P2, P0, like that. P1 and P2 are used for the transcription of stable X-cyst transcript, and P0 is usually meant for transcribing an unstable X-cyst. So based on which promoter is being used, the stability of X-cyst can vary. So P1 and P2 usually gives a stable X-cyst, P0 gives an unstable X-cyst. So you can guess it. So the paternal imprinted inactivation, most likely it will be the P0. Why? Because P0 is not very stable. So after some time it will be unstable, and once the promoter is off, that chromosome can be revived back. But whereas later stage in the organism's body, it has to be P1 or P2. Why? Because you want a stable X-cyst so that that chromosome maintains. So you don't want a weak X-cyst. 2.12p region shows H4, means histone 4 hyperacetylation in undifferentiated female embryonic stem cell. So this is a region like P arm and Q arm are the 2 arms on the either side of the centromere of a chromosome. 2.12 region of the P arm will show a histone 4 hyperacetylation in undifferentiated means it is maintaining stemness or stem cells, a female embryonic stem cell, and hyperacetylation disappears upon differentiation. Once the tissue decides to differentiate, like stem cells can be forced to differentiate into neurons and other tissues easily by different chemical cues, but they will have this hyperacetylation disappearing once the differentiation kicks in. Stabilization of exist RNA might depend on the levels of exist RNA present. So amount of exist RNA also can contribute and decide whether or not an exist RNA must be stabilized or not. Like you are going to let us assume a situation like you are going for a war. Only one person or two person are there, you will not be brave enough. So your stability can be in question. But if you are going 100 people or 500 people or 10,000 people going, then you will have strength in number and automatically it will enhance your chance of survival and stability. So this happens in the case of exist RNA also. Their number also depend, the load or the amount present also can contribute to the exist stability. So now let us see in a cartoonish manner how X chromosome inactivation is taking place. So this is the X-Inactivation center and the central point is the exist RNA itself. Once the transcription has started, we know that there is histone modification that kickstarts, that is deacetylation takes place, hypermethylation takes place, etc. So this spreads like a nucleation point right from here, and they also contributed by certain repeat elements such as lines, long interspersed nuclear elements. So lines and signs stands for short interspersed nuclear elements. They are repeats, genome repeats, they are present and they can contribute as stations for spreading this inactivation. So it is spreading it is spreading like a wildfire. Normally in forest, if there is wildfire, it doesn't start from everywhere, it starts from a center and spreads. Like if you see satellite or aeroplane pictures, you will see wildfire spreading like circles to start with, and the circle become bigger and bigger and bigger and can spread across. So like that it is spreading like this across the entire chromosome, and not that this chromosome was longer, now it became shorter because of the compaction, and exist RNA start coating now onto the RNA, and which in turn will attract more heterochromatinizing enzymes, that is establishment of inactive state and asynchronous replication takes place. That means this X chromosome, the DNA of this X chromosome is now asynchronous to the rest of the DNA, rest of the chromosome, means they became like out of sync. It is not in synchrony with rest of the chromosome, so it is following a different route. It is following very slow replication, very late replication, etc. Credit goes to this compaction. And finally you have the recruitment of this macro H2A histone isoform, and histone 3 and histone 4 gets hypoacetylated, means deacetylation takes place, and that will make the chromatin very tight as you see here. Let us understand more about the T6. T6 is transcribed from the antisense strand relative to exist. Exist is in one strand, so exist is in the top strand, T6 is in the bottom strand. So in undifferentiated cells, T6 is expressed in both X chromosomes. So in undifferentiated cell means it's not committed to become anything, so T6 can be expressed from both. In differentiated cell, it is present only in active X chromosome, that is written as XA. So active X chromosome must have strong expression of T6 to counter even the leaky expression of exist. Even if T6 is expressed, that doesn't mean that exist cannot express. The moment exist express, that also will be vulnerable for compaction. So T6 makes sure that it do not allow any exist to coat onto the DNA. So it will prevent the upregulation of exist on active X chromosome XA. So exist should not be expressed in T6 expressing chromosome. and it influences X chromosome choice and imprinting. So T6 also can decide whether or not a given X chromosome should be active or inactive. So it can decide the choice of chromosome for the imprinting purpose. So let us see how the transcription of X-cyst and T6 takes place in a cartoon fashion. So here you can see T6 is transcribed, and you know the outcome. When T6 is transcribed, it can create the masking effect. It can cause a masking effect onto X-cyst, and then if T6 is expressed, it can create an interference. Remember, when the transcription has to happen, the transcriptional machinery is assembling onto one chromosome, so it can create steric hindrance. Like you are entering a room and another person also is coming out of the room in a bus, rather, that will be a better example. Door is smaller, one fellow is getting out and you are getting in, so there will be a stalemate. Neither that fellow can come out nor you can go inside. In train, bus, you can see it. So this is called interference. It is kind of interacting, and of course here interference means it can pair and it can cause the degradation of both the strands. And then comes repression. What is repression? The X-cyst promoter is inhibited by the T6. So this region where the T6 expression is, its regulatory region, can inhibit. So they can sequester and titrate away all the elements required for like you may have seen in some movies, etc. Like there may be one shop, one small shop where he's selling juice or something, and then if a posh shop, very nice looking attractive shop comes in the neighborhood, then everybody will start going to that new good looking shop. The small shop fellow will lose business. Something similar, that there will be more attraction happens to a given promoter. The other promoter is lacking. It is not having enough nutrition in terms of what you call transcription machinery. It will be sequestered by this inhibition, and then also by chromatin opening. Whether or not the region responsible for the transcription of X-cyst is allowed to open. First of all, chromatin has to be removed, or chromatin has to be loosened, or chromatin has to be remodeled. That is called remodeling of chromatin for accessing the DNA for this RNA polymerases, so that the gene expression can take place. What if this compaction doesn't happen? You are not allowing this unpacking at all. Then also the X-cyst chromosome cannot express. So let us see what are the mechanisms. Masking can be done by T6, and interference can be done by T6, repression can be done by T6, and chromatin opening can be inhibited by T6. So 4 different mechanisms are possible that can interfere with X-cyst expression of and vice versa. You should also think that T6 is expressed, got an upper hand. The same mechanism can be done by X-cyst also, because X-cyst expressing cell should not be able to, or X-cyst expressing cell don't want a T6 promoter to be expressed active in them, because X-cyst expressing chromosome want to be inactive throughout. And one more thing is that in a given cell, if one paternal or one maternal, either of them are inactive, it will continue throughout the lineage. If that cell is giving rise to 2 cells later and 2 becomes 4, in their continuous lineage, it is not going to be reversed. It will continue as long as the organism or that cell is alive. It is not going to get reversed. So it has to be decided during the early developmental stages itself, the randomness. But it can happen sometimes. A given paternal copy or maternal copy expressing cell do not survive for whatsoever reason. Then what will happen? That space will be occupied by the other cell. But this will usually do not happen the way I am saying, because once a given X chromosome is inactive, all the subsequent cells coming from that particular cell will continue to have the same chromosome maintained in inactive state. So maternal is inactive, it will stay like that. Paternal is inactive, it will stay like that in that particular cell and its downstream molecules. So you can also see this cartoon what has been shown here, how the mechanism is working for the inactivation purpose. So we can see how X chromosome inactivation and how it is influencing the RNAi. So X chromosome inactivation is a good example of RNA interference. So X-cyst and T6 can be taken as a typical example of RNA inactivation or RNAi itself. So we will study more in detail about the X chromosome inactivation, how the RNA interference comes into action in the inactivation process, and we also see some pictures, images that explain that the actual strength or depth of inactivation with some RNA in situ images in the next class. Thank you.